Hi, everyone, and welcome to Cytobank's webinar on citrus training and advanced applications. My name is Geoff Kraker, and I'm an application scientist here at Cytobank. Also joining me later on this webinar will be Kat Drake, our Director of Informatics. She is kind of the force behind citrus here at Cytobank, and we are happy to have her along to answer some questions. Just getting started, in case you're unfamiliar with Cytobank, I'm sure a lot of you have at least seen Cytobank before, but if you're just joining us, Cytobank is a web-based platform for data management, high-dimensional analysis, and archiving. So we have a couple different pieces of our software in our company. The first, like I mentioned, are high-dimensional analysis tools. These are things like Citrus, Spade, and ViSNE. These are useful for looking at really complex experiments. And in addition to that, in our software, we have the structured content management. And these are tools, like I said, to archive or to collaborate with other scientists. And those comprise our software offering. But in addition to that, we have our informatics consulting services, which Dr. Kat Drake is essentially in charge of. And these are where we do bespoke analysis using tools inside and outside of Cytobank for customers on existing data sets. So I won't talk much more about this. Again, this is a presentation focused mostly on citrus, but you will hear this mentioned one or two more times. So what is citrus? Citrus is an algorithm that is used for the identification of differential single-cell biomarkers. So here's the paper, which kind of debuted the algorithm. And essentially, this is the workflow. So you start with a scientific question and single-cell data, and then you enter that information into Citrus. So this is Citrus is technically a couple of algorithms put together and kind of inside of we call it a black box approach. So you don't have to know how to do any of these things on your own. The algorithm does them all automatically. The automatic identifying of cell subsets, that characterization of those subsets, and then a regularized regression testing on those. And then Citrus presents to you kind of these stratifying biomarkers, telling you kind of what markers lead to the differences between your two groups. So Citrus is used kind of on a large scale that lets you compare groups of samples and tells you which differences between those groups are significant. So the workflow that we're going to be talking about today is essentially as follows. We're going to talk very briefly about the raw data processing, just making sure you're starting with quality data. Next, the workflow involves running ViSNE first on that clean data, then running Citrus multiple times on the result of that ViSNE analysis, finding your differences between your groups, and then looking at those differences in Cytobank back on that ViSNE map. So this allows you to do essentially something very similar to this, where you have groups of data. It could be anything, but responders or non-responders, or treated and untreated, whatever you'd like, and you can see these ViSNE maps. They're basically the same for that single experiment. And then you can take the significant citrus clusters and map them back to that ViSNE map. So it gives you context for where your significant clusters are. In addition to showing these clusters, these cells that comprise these significant clusters of groups, you can also show, kind of more traditional ViSNE data, where you can look at the expression levels of different surface markers or functional markers displayed on that same ViSNE map. So it kind of gives you this, like I said, way to link all of your information together with context. So what are the steps to running this pipeline in Citrus? First, you need to define the comparisons you want to make. You have to think about what you have, what data you have, and how you want to compare it. Next is kind of pre-gating and data scaling, making sure that the data looks good. Next, run ViSNE on that original experiment that you've kind of curated a little bit. After that, run Citrus multiple times on that same ViSNE parent experiment. This gives you a way to compare across multiple citrus runs. After that, interpret the significant clusters output by citrus. Like I said, map them back to the ViSNE map on that same region. And then compare that information to how you might manually gate for a population, or look at those citrus clusters and say, okay, how would I develop a gating hierarchy to get to these based on two-parameter dot plots? And that kind of essentially is a whole workflow that we're going to be covering today. So the first step is understanding the comparisons you want to make in the data. So in this case, like that example I used before, there are responders and non-responders, and this is the main comparison variable. This is, in this case, the thing that you want to compare the most. But you also might have other stratifying variables like time point 1 and time point 2. So if you want, put all of the files from one stratifying variable into one Cytobank experiment. So it helps to break this down as much as possible to help Citrus's statistical power. But there is a caveat here, actually, and it's important to mention. So you should keep paired samples in different stratifying groups. And what's the takeaway message here? What does that mean? So if you're looking at the same patient over time points, pre-treatment, post-treatment 2 weeks, post-treatment 6 weeks, etc., those time points should be in separate stratifying variables. So you can still compare them, you just can't compare them in Citrus automatically. And the reason is that Citrus operates, it's not exactly the same as, but it's comparable to kind of a standard t-test. And if you're looking at differences in the same patients between time points, you would use a paired t-test. And there's not a quote-unquote paired t-test version of Citrus. So it would be inappropriate to put different time points as your different groups in Citrus right now. However, we can still do the comparison later on manually, and I'll talk about that. So Citrus is fantastic for looking at groups, differences in groups like responders and non-responders. But again, keep your stratifying variables separate if they have the same patient across those variables, or the same mouse, or the same whatever kind of sample you're looking for. So after you've chosen how you want to compare your groups, how do you kind of clean up the data a little bit? First, at least pre-gate to singlets. Doublets look like small, interesting populations to the algorithm, and so it was going to pick those up, certainly. Across other algorithms in Cytobank as well, ViSNE and SPADE, doublets just essentially make the analysis more complicated. So at least gate to singlets, obviously live cells or 45 positive if you're working within kind of an immune cell framework. Those would be good places to start. However, you might want to pre-gate further if you want to focus on a more refined population. If you only care about Tregs, you might start with just your CD4 positive T cells. You can kind of get rid of the monocytes and the B cells and the NK cells and stuff. So this is a good place to start. However, you also may need to combine many samples in your ViSNE and detect rare populations. So if you're looking for a lot of rare cells, or you have many samples, you'll need kind of some additional headroom in your ViSNE, and I'll talk about that in just a second. All channels that will be used for the analysis should be transformed so that the distributions are bimodal or normal, and that the scale ranges should be adjusted so that you can see the data actually on scale. And this is just a good standard operating procedure anyways, but just make sure all your data, if you view it on a two-parameter dot plot, that most of the events, almost all the events show up on scale. They're not pressed up against the axes. This is important, and this point was made, that they should be transformed so distributions are bimodal or normal. If you use other data other than flow cytometry data, Citrus will work for any single cell data. So if you're looking for segmented image files or looking to run Citrus on single cell RNA-seq data, for instance, just make sure that your data is distributed like that. So after you've checked your scales, you've made sure you know what you want to run Citrus on, how you want to compare your groups, The next step is running ViSNE on that clean data. So use the same channels for the ViSNE clustering that we'll use for the citrus clustering. So these are generally stable surface markers. And we can talk more about that later. And please feel free to ask questions kind of during this presentation. Like I said, Kat will be around for a Q&A session at the end. And I would encourage you to take advantage of that. However, after setting up ViSNE with those surface marker channels, essentially the channels you want to use to separate your cell types, then feel free to optimize the ViSNE map for visualization of the results to make the downstream comparisons easy. So basically, this just means tune your ViSNE controls to make the map look better. And feel free to run ViSNE a couple times with a couple different kinds of settings to make sure that your map looks good. So as you add more events, it's a good idea to increase the number of iterations and the perplexity in the ViSNE. So like I said, try a few different settings in ViSNE to see if you can find one that generates a good map. So I won't spend too much time talking about this because this is a Citrus webinar after all. But again, the perplexity variable, or the perplexity setting in ViSNE is set to 30 by default. If you increase that, it tightens the islands kind of around their center a little bit. So this can be helpful if you have kind of smudgy data. If you're running something like bone marrow where there's a lot of intermediate states and immature cells, this can help give some definition to the ViSNE plot. Also, if you have ViSNEs that look like this, where there's kind of swirly patterns in them, and it would be really difficult to kind of draw a gator on this, this is an indication that you don't have enough iterations. So feel free to increase the iterations on your ViSNE settings as well. So the reason to run ViSNE, again, I've covered this a little bit, is so you can always map your significant citrus clusters back to the ViSNE map for comparison. And so this is what an experiment might look like. You might have arm A and arm B, and you would compare those with citrus at baseline, compare them after treatment during the monitoring steps later on. So these would be 4 independent experiments in Cytobank running citrus on the comparison between arm A and arm B. But this can add up even in these. Obviously, we recommend with citrus you have at least an N of 8 per group, and so baseline you're looking at 16 files. If you're looking at rare events in those 16 files, they need to be relatively populated with cells, and so ViSNE and Cytobank, and this is an important distinction to make. ViSNE and Cytobank can have up to 2 million events per run. So that's essentially your cap for the number of events in this workflow. If you have the need to run more than 2 million events in a ViSNE run or in your resulting citrus after that, we'll talk about that in just a second. So essentially, you want to run a lot of files on your experiment through your ViSNE in order so you can have enough samples for your citrus. So in order to know how many events you need per file, you can use this little kind of helpful chart. So if you're looking for an event that's 1 in 20, that's 5%, that's about a B cell, plus or minus a little bit. You need to see, with a desired CV of 5%, you need about 8,000 cells per sample. You need 16 samples, so it's this times 16, and that's very doable with ViSNE. However, if you're looking for a cell that's 1 in 100,000, and you want a relatively low CV, all of a sudden you're looking at 40 million cells, and that times 16, obviously, is far, far, far beyond what can fit in one ViSNE run. So if that's the case, like is kind of outlined here in this little flow chart, you should consider pre-gating further, like I talked about at the very beginning, making sure that you've trimmed all the excess populations of cells that you don't care about out of your starting ViSNE, or meta-clustering, so basically clustering on clusters. And this is a workflow that's not available in Cytobank, the software alone, but is offered by our consulting services. So I'll have some contact information up at the end about how to get in touch and reach out to us and start talking about kind of this workflow and how we might accomplish that for you. but just keep that in mind. It is possible to do kind of larger scale comparisons if you need more than 2 million events. So now you've got your question selected, you have your data cleaned, you've run ViSNE on your data. What do you want to do now? So you have kind of this branching choice to make. So to run CITRUS, there's kind of two different versions of the algorithm. There's the correlative CITRUS, which is if you want to look for all significant differences that your panel can measure. Or there's the predictive CITRUS, which is if you want to find the fewest number of markers needed to predict an outcome. And this is more of a, correlative CITRUS is kind of a more hypothesis generating, like I just want to see everything that's different between my groups. And then predictive CITRUS, the reason you might want to find the fewest number of markers to predict an outcome is if you're looking for a specific biomarker, or you want to make maybe a sorting panel to narrow in on one way. So you're using a lot of parameters to find one, and then you're using that one in a panel to sort your populations apart from each other. So there's kind of two approaches here, and we'll cover them both in this webinar. So correlative CITRUS. What does this mean? There's also, in addition to predictive and correlative, you can look at two different things. You can either look at the differences in medians between your groups, and this is if you're looking at a functional marker, something like a phosphosignaling marker or a set of phosphosignaling markers, or if you want to look at the differences in an excitation marker or an exhaustion marker. A lot of people are doing work in those these days. So if you want to tell the difference in the median of a channel or median of several channels, you can select this version. If you want to see the change between your groups in abundance of a population, if you want to see if your Tregs go up or down, or your MSDCs go up or down, these are, you would run that in the abundance mode. So you kind of have to pick, in addition to like I said, predictive or correlative, you have to pick if you want to look at the change in a population or the change in the median of your channels. So if you want to run correlative CITRUS after ViSNE, split your files into experiments by your stratifying variables. This is what we talked about at the very beginning. Run ViSNE on everything, and then split those files into those separate time points into those experiments, and that's possible inside Cytobank. And then after you have everything split by stratifying variable, run SAM, which is, it stands for Significance Analysis of Microarrays, but that statistical method has been kind of adapted for use with proteomic and flow cytometry data. So run SAM at least 3 times, and this is kind of a theme you'll hear me repeat a couple times in this webinar. It's important to repeat CITRUS to make sure that you are not getting any false positives. So at least 3 times is enough to make, convince yourself. You're like, all right, cool, I think I'm onto something here. If you want to bring it to your lab meeting, and tell your boss about it, maybe run it 5 times. And obviously, the more sure you want to be about it, feel free to repeat CITRUS as many times as necessary. But at least run CITRUS 3 times. And this is the same is going to be true for the predictive version as well. In that case, we'll say run PAM and LASSO at least 3 times. So just baseline, take these settings, and there's a link in Cytobank. It says copy analysis settings. You can click that to quickly set up more duplicate CITRUS runs. So you do that after you have, this is essentially the minimum cluster size. And this is this should equal the estimated abundance of your population of interest, your smallest population of interest in your starting population. So if you're starting with CD4s and you're looking for Tregs, the minimum cluster size will be the abundance of your Tregs in your CD4s. If you don't know what your minimum population of interest is, your smallest population of interest is, you can skew lower. However, don't just automatically put in 0.5%, because then it's much harder statistically to detect significant differences between your groups. So it's a balance between picking up your smallest population of interest and also not washing out all your significance by doing so. So again, feel free to change this value if you don't know what your smallest population of interest is. enter in a few and see if you can pick something up. The next setting to change is set the CV fold equal to 1. Traditionally, this setting in Cytobank dictates how many times it does a cross-validation step. However, a CV fold setting of 1 basically is a special case. That means it just does this cross-validation on one section of the CITRUS algorithm and not everything, so it ends up being much faster. And this is more appropriate to run with SAM if you're doing predictive. And I'll talk about this a little bit. We're going to want to increase this to higher. So lastly, in the setup of the correlative CITRUS, you want to normalize the scales if your dynamic or absolute range of channels varies by about 5 times. This is much more common in flow cytometry data. CyTOF data, the dynamic range doesn't change that much. However, it's okay to turn this on by default. You can run it all the time, and it won't have a detrimental effect on your data. So if there's some question, if you're like, I don't know, feel free to turn this on. Just as a quick sidebar, and I think Kat's going to address this later on, are the results of CITRUS significant, and what's the p-value of these results as we get into them? All CITRUS results are statistically significant below a false discovery rate threshold set by you, the user, and this is variable. For the correlative version of CITRUS, it generates 3 basically separate results. One with an FDR of 1%, 5%, and 10%. And this is it's similar to a p-value adjusted for multiple hypothesis testing. So it's similar to a p-value of 0.01, 0.05, and 0.1. So you can report your CITRUS analysis. Sometimes we get questions of like, how do I technically say what I've found with CITRUS? So you can say these results are statistically significant below a false discovery rate cutoff, below whatever, below 1% or 5% or 10%. Sometimes, this is just part of statistics. There is a 1% chance or a 5% chance or a 10% chance. One of those findings will be a false positive, depending on what version you're looking at. So this is why we say repeat at least 3 times. It's to make sure you have stable results. You're looking for the same populations being found in each repetition of your CITRUS run. So predictive CITRUS, this is the other branch of kind of the decision tree you have to make. So if you want to find the fewest number of markers needed to predict an outcome instead of everything different, you can run predictive CITRUS. And that's using the PAM and LASSO association models instead of the SAM association model. So again, like I said, run PAM or LASSO at least 3 times, just like the correlative CITRUS. You can set the false discovery rate to whatever value you want within a range for the predictive version of CITRUS, not just 1, 5, and 10%. 5% is a great starting point. That's pretty common statistical practice. But if you want to be more rigorous, if you want to, have that much less of a chance of finding a false positive, feel free to type in 1%. And again, these are kind of similar to a multiple hypothesis testing adjusted p-value of 0.01 or 0.05. Same rules apply for the minimum cluster size. If your estimated abundance of a population of interest, if you know that, feel free to enter that in. If you don't know, skew a little bit lower than you estimate, but again, don't skew too low because you're kind of making it much more difficult to find the any significance any significant differences between your groups. In this case, for the CV fold, it's a good idea to make this equal to the number of samples you have in each group, or as high as possible. So if you have only 8 samples in each group, and that's kind of where you can start running CITRUS and get significance out the back end of the algorithm, you'll only be able to set this to 8. However, if you have more, if you have, for instance, 20 per group, you only need about 10 CV folds. So set it to 8 if you only have 8 samples, or 10 if you have more than 10 samples. And again, the same thing. It's okay to normalize the scales. It's okay to leave that on by default, but you definitely should turn it on if you have different dynamic ranges of your channels. And just so I can give you guys an example, this is, for instance, if you're doing flow data and you have, for instance, PE is a very, very, very bright channel, and Pacific Blue, or there's some other dim ones out there, are very dim, and they differ. A positive in Pacific Blue and a positive in PE will differ by orders of magnitude in how bright they are. And if that's the case, then you should normalize your scales. So how do I interpret these results? And here we'll kind of take a step into general interpretation of both models, and specifically in this case for the predictive model. So how do I look at the results now? So after you've run these, after you've changed the settings to find your minimum cluster size, and then repeated that citrus 3 times or 5 times, what do you do with the results now? So if there's no model output returned, if there's a blank folder, that means there's no predictive results. That means it didn't find anything. There's no predictive differences between your groups. However, for the predictive models, you will find kind of one more PDF in your results. And again, if you're unfamiliar with the citrus results, instead of being more channels like ViSNE is, or having a viewer in the software like Spade does, the citrus results are essentially kind of a little package. It's a zipped package, and it contains a series of PDFs and some spreadsheets. And you get an extra PDF in your predictive results, and that's the model error rate plot. And this should look good, in quotes, when if you do get results, you should first look at your model error rate plot to ensure that it looks good. And I'll show you what that is. I'm sure a lot of you are like, all right, so what does good look like? There's 3 different versions of a predictive citrus result as well. There's one called CV-min, one called CV1SE, and one called FDR constrained. And these are all visible on the model error rate plot. So just FYI, just so that you have like a written rule, the CV-min or CV1SE should have fewer features than the FDR constrained model. And an FDR constrained model should be found. So what does that look like? So here is a model error rate plot that looks good. You can see there's a little a key up here. This red line is the cross-validation error rate. That's essentially, it represents during the cross-validation step in citrus, it takes a subset of the data out and compares it against the model that it's building to make sure it's a rigorous model, to make sure it's not building the model to fit exactly only your data and that anything else would completely fail. So it takes a subset of your data out and compares it against that model, looking at kind of a series of model features. And these are either the abundance of a cluster or the median value of a cluster. So either one of those could be a feature depending on what you started with. And so it says, all right, just looking at these 2 features, do I have a good model? And it fails about 20% of the time. So it moves on to 2 more features, then 2 more, and then starts adding more features to the model that it's building. So when it adds about 3 features here, it looks like then it starts to become a better model because the model error rate drops. So it more accurately predicts the outcome each time. So that's a good thing. So it's a good thing to see your model error rate, your cross-validation error rate drop down. You want it to be as close to 0 as possible. However, it doesn't have to go all the way to 0. There are, depending on your scientific question, a model error rate of, a cross-validation error rate of 40% might be good. If you're looking, the reason you're using citrus is to see if you can predict if a patient will get diabetes in the next 5 years. And if you can do that 60% of the time correctly, that's fantastic. So if your model is wrong 40% of the time, you know what, that's okay. It's a little high, but it's still pretty good. It's better than what we have currently. However, if you, for instance, on our demo experiment that we have on all of our servers for citrus, is just stimulating B cells with B cell receptor crosslinker and then looking at phosphosignaling outputs, that's a really strong signal. It's really easy to tell the difference. So the cross-validation error rate will vary. It doesn't have to go down to 0, and something as high as 40% might be okay. So just keep that in mind. It depends on your scientific question, whether or not you'll be happy with the cross-validation error rate. But in general, it's a good idea to see that drop over time. The feature false discovery rate, this blue line represents essentially the time where if you add more and more and more features, eventually one of these features is going to be a false positive. And this blue line is a representation of that. And so the CV-min and the CV1SE are showing you basically the model with the fewest number of features needed to show significance between your groups, a significant difference between your groups. The FDR constrained, this little icon here, is essentially representing all of the differences between your groups before this false discovery rate line rises above what you set your threshold to be. So in this case, we set this threshold to be 5%. So when this blue line crosses 5%, that's where the FDR constrained gets placed. And so basically what you want to see is you want to see these two, CV-min and CV1SE, have a fewer number of features than your FDR constrained model. You want to see your blue line start off relatively low and then increase above your threshold, and you'd like these to be before your blue line crosses that threshold. So here are examples of bad model error rate plots. In this case, you can see that for this first one, the feature false discovery rate skyrockets all the way up to looks like 30% before either of the CV-min and CV1SE exists. There is no FDR constrained model. That's a limitation. But this one, it looks bad. Also, this one looks bad. There's still no FDR constrained model. The feature false discovery rate is crazy high to begin with and never drops, below about 40%, which means basically there's a good chance everything is your models, everything your model predicts is a false positive. And this red line doesn't drop down, close to 0 at all. So these are both bad plots. This one, however, is kind of borderline. You might find things like this. Not everybody's model error rate plot is going to look like this. This is an example of a very, very good one. Most people's will look, kind of plus or minus this one a little bit. So, obviously it depends on the data you're using and if there's a large change or a small change. But this one isn't necessarily bad. So again, the cross-validation error rate is kind of high. It looks like it's about 35 or 40%. However, if again, depending on your scientific question, it might be totally valid. It might be okay for it to be that high. There is an FDR constrained model, and it is before it has more features than the CV-min and the CV1SE. So this isn't necessarily bad, although it's a case where I definitely run Citrus five more times to see kind of what the results will look like. So how do you interpret Citrus for the correlative and predictive modes kind of on a macro scale? So the first step is, do I have any predictive results at all? Do I have any results? Like I said, if there's no results, basically you won't have PDFs in your folder. So that's a good indication. If that happens, either you have a change that's so small that it's not visible to Citrus, and you need more samples per group, or there may, honest to goodness, be no real change between your groups, and adding more samples won't do anything. Next up, though, are my results able to discriminate my groups? Can it tell your groups apart? And are the results also significant? Next step, you say, okay, so I have significant results that can tell my groups apart. I have the results to begin with. What am I? Are they stable and consistent? So, is the general level of predictive accuracy stable across the repeats? So, if I've run Citrus 5 times, do the model error rate plots look similar? Is there a similar cross-validation error rate across those repeats? Is there more than one set of equally predictive markers? And you can use the ViSNE map to tell this, but essentially, it might be that your model has 3 different phosphosignaling markers, for instance, that are equally predictive for the difference between your groups. So if you get different markers but the same model error rate, that's okay. It might just mean that one marker is just as good as the next marker at pulling apart your groups. And the next, after you determine they're stable and consistent, and again, you use the ViSNE map to tell this, you map your significant clusters from Citrus back onto the ViSNE map so you can see if they are mapping to the same region over and over and over again. And then, like I said, the last part, what are my results? So what's the phenotype that's predictive or of these predictive populations? And then what difference between the groups is predictive? So these are the questions you kind of have to use to kind of guide you through analyzing Citrus in this version. So first up, do I have any results? Like I said, if no model was returned, there's no significant results. 5% is a great false discovery rate to start with. Your results from Citrus will appear as a series of PDFs, and I mentioned that. I'll kind of walk you through what they look like. So identify the significant clusters on the features plot. So choose the one. You'll get this features plot, features CV-min, features 1S, CV1SE, or 1%, 5%, 10% for correlative. What this plot means is kind of just a binary. It's saying, all right, with Citrus, this node in the middle represents all of your events in your Citrus analysis. And as it branches out, it's separating your cells by phenotype from all of your files in your analysis. So what this is telling you, and you can see these are tiny little arrows, so it's kind of this kind of branching effect. It's different than a spade tree. These are nested, so if this was a population, this would be parent, this would be grandparent, great-grandparent. It's hierarchical, kind of like a gating tree. And so what's being shown here is if there's a red node, that means the events in that node are either predictive or correlated with the differences between your groups. And if there's 2 kind of in a chain or more in a chain, that means the grandparent population is predictive, the parent population is predictive, and the children are also predictive. However, you should choose, if you're doing this kind of workflow to map back to ViSNE, you should choose the highest bubble in that tree, or the one closest to the trunk, if we're extending this analogy further. So you should use that population and export that population for examination. So this node, because all of these are nested, this node contains all of the events in these other nodes as well. And same with this one. These are not nested, so this one is kind of contained inside this one. So once you have significant clusters, you can then export these clusters to a new experiment along with the original files. You can concatenate the significant clusters from each of your files together and your total files together, and then upload them again to a new experiment, and you can overlay them. And I'll show you what that looks like in a second. So this is essentially a workflow describing that. So export events split by comparison variable and Citrus cluster, or basically all cells. Concatenate the events with annotation channels for comparison between variables and Citrus clusters. Use the color overlay plots with the cluster gates to show significant clusters on the ViSNE map for each replicate. So again, I'll show you what this looks like and talk a little bit more about this. And then use the color overlay plots with the cluster gates to show significant clusters mapped back on ViSNE within each stratifying variable. So again, remember we ran ViSNE on everything. We broke apart the files by stratifying variables, so you're not running CITRUS on time points or other similar things. But you still, the reason why we run ViSNE on everything is you want to start with the same ViSNE map. So even across stratifying variables, you can map things back to the original ViSNE. So the CITRUS clusters identified similar cells across replicates at each time point. So in this case, this is from our demo data set. There's one time point, but this is 3 replicates, and they're all very similar because, again, I mentioned the data is relatively simple. It's slamming B cells with B cell receptor crosslinker and looking kind of at the phosphosignaling fireworks. So it's really easy to tell the difference between B cells. So this little island are the B cells, and there's 3 separate clusters from 3 different time points you can see here, or sorry, 3 different replicates. Essentially, we've been working through what are your results. You can use multiple tools. So the one that I've been talking about, and we'll show a few more examples of, is this ViSNE map colored by marker expression. So you can see kind of just what cell types and how they behave are, kind of across all your ViSNE islands. You can see the citrus tree colored by marker expression, so you can see how citrus has decided to break apart your cells and in their cell types. And then histograms generated by citrus for the significant clusters. So you should just ensure that the significant clusters generated by citrus are relatively homogeneous in their marker expression. This is, again, making sure your results are consistent. Use the box plots that citrus generates to look at the magnitude of the differences between your groups. Again, it's great if citrus finds the same magnitude between the same clusters. But just remember that as abundance, because it's looking at essentially percent, if one cluster goes up, other clusters will go down, kind of as a rule, just across your total cell population entered into citrus. So ViSNE maps, these are colored by marker expression. This is that first one I talked about. So instead of looking at just these overlaid clusters, you can display that same ViSNE map colored by marker A. In this case, this is CD20, or marker B, this is CD4. So this is basically a way to get some more context on what's going on. This is the citrus tree from this analysis, colored by marker expression. So in this case, this is CD4, this is CD20, and this is HLA-DR. You can see there are 3 significant clusters here, green, orange, and red. They're all kind of nested. That's why they're all overlaid on top of each other. And that's these significant clusters here. Each of these clusters has a little number inside of it. This is an ID tag. This is so you can choose specific clusters to export as part of that workflow like I was talking about. So next up, you can see for those 3 significant clusters I was talking about, you can see histograms of their surface markers. The blue histograms are the expression levels of all cells, and the red is the expression level of just your significant clusters. So in this case, you can see that our significant clusters are CD3 dim or low, CD4 negative, and CD20 bright positive. So we get confirmation that sure enough, those significant clusters are B cells. And then for each cluster, you'll see this basically box plot showing the differences in transform median in whatever particular channel it was different in. In this case, it's phospho-S6. So you can see the differences in signaling between everything else. So these are, these are our kind of unstim samples and our stimulated samples. So again, you want to make sure that the accuracy is similar across repeated citrus runs. And like I mentioned before, I just want to reiterate this. It's okay if the biomarkers returned are different, but result in a similar accuracy. That's okay because it could be that they're equally predictive of changes between your groups, the differences between your groups. So after you've gone through and gotten these populations exported from Citrus, these significant clusters, you've concatenated them together and uploaded them back to the same experiment as kind of the parent files, and you map them back onto that same ViSNE. You say, all right, great, it's mapping to the same regions. You can now compare them to manually defined populations. So you can import manual gates into your Citrus exported clusters experiment, and then basically look at your clusters and kind of backgate to see how your clusters map to your kind of traditional gating strategy, to understand kind of where they fall, in your manual gates. So this is an example of what that looks like. So this is kind of on the ungated data. These are, it's CD20 and IgM. And you can see here that for these, significant clusters from Citrus, these are the kind of responding B cells. This was my manually drawn B cell gate, and this is where the Citrus clusters map to on that. And that's okay. You'll see some events kind of fall outside of where your manual gate might be. In real life biology, things are normally distributed. Things don't stop at a hard edge. And in fact, this is actually doing much better. You can see some kind of slate blue events in here. My manual gate would have picked those events up. And Citrus, because of the way it's structured, discriminates and says none of those, even though they fall within what I might, as a fallible human, define as a B cell. Citrus says no, they don't behave like B cells and they don't look like B cells. So if you look at all the markers at the same time, so it didn't put them into these populations. So if you want to see how Citrus has been used in other publications, here's a reference. If you say, hey, this is great, and your boss is like, but how have other people used that? Here's a reference from Ben Shannon et al. In 2016 in Nature Medicine. Basically, they looked at a mouse model that has an inducible placebo response. And then they looked at basically the immune system in these mice. And they found, using Citrus, that when you induce a placebo response, those mice with this placebo response actually had basically found that functional immune markers were upregulated in immune populations within that induced group, as opposed to the controls. So it's kind of a fun model. It's an interesting use, and also they successfully use Citrus to show their result. So definitely check it out. So now we're going to have time for a Q&A. However, I'll leave these links up during it so you can have access to these, and we'll have a recording of this webinar up in the next day or two. And so please feel free to view it again or show it to people, lab mates who missed this. You can check out our YouTube channel. This is where it'll be hosted. Contact us at services@cytobank.org to schedule a consultation for our consulting services. We'd love to sit down and talk to you if you're interested in things like meta-clustering or using tools, in addition to Citrus, tools available outside of Cytobank on your data. We can certainly help with that. And lastly, if you're interested in seeing kind of Cytobank work in real life, or kind of having me kind of walk through Citrus, please contact sales@cytobank.org to set up a demo. So with that, I think I'd like to bring Kat on board, and we have some questions stacking up here. So if you have questions and you're holding them until now, please enter them in the Q&A section, and we'll work through them. We've got some time, but if we don't get to your question, we'll by all means certainly follow up with an email. All right. So Kat, welcome.
Hello, everyone.
So we have a question here. First up, well, we have several, but first is, What if my outcome isn't a group? Can I use citrus with outcomes like continuous response measurement or survival?
That's a great question. That's one that we get a lot. Um, so right now in Cytobank's application, you have to define, as Geoff went over kind of at the beginning, you have to define your outcomes as groups that you can compare. So one way to handle it, if you have a continuous outcome or a time to event outcome like survival, is to find a way to define that as groups, like put a cutoff on the continuous response, for example. But we also do have custom pipelines that we build sometimes using our Cytobank's application programming interface or API to help our customers with those kind of analyses and services. Yeah.
Great, thanks. So David asks, could you repeat why not having an FDR constrained result in your model is a bad thing?
Yeah. So Geoff was talking about that in reviewing the model error rate plots for the predictive models. And what we're doing there when we look at the three different types of constrained models and compare the FDR constrained model to the other ones is we're basically using the FDR constrained model to make sure that there is some significant signal in the data after accounting for multiple hypothesis testing or after accounting for the fact that we're looking at so many different clusters and testing them for significance. So if the algorithm is not able to find an FDR constrained model, that is an indication that there's not a lot of significance in your results and means that the other models that it's returning might not be very likely to work if you take that predictive model into new data sets.
Here's another one kind of related to citrus repeats. Lauren asks, can you elaborate what you mean by repeating citrus results 3 to 5 times? Do you use the output from the first citrus run on the subsequent ones? Do you combine the citrus repeated outputs as a final data set or choose a representative run?
That's a great question. So what we do in this case is actually just literally repeat the citrus analysis the exact way that it was run the first time. And you can do that in Cytobank by setting up your first citrus and then hitting copy analysis settings and just hitting run again without changing anything. And then we use those ViSNE maps that Geoff was showing to compare those results and make sure that they're consistent. In the end, for a final data set, we typically will just choose a representative run. But we also do have some more advanced workflows to look at repeating citrus that we do in services, again, using the API in contexts where we really want to be really sure and dig in and kind of understand the results better. So just to give some kind of examples of those sorts of things, one thing we do is repeat the analysis hundreds of times and track when specific cells are in significant clusters so that we can measure the probability of that cell being significant. We compare that to random scrambling of the outcome data to see how that's significant. And then another thing we sometimes do is run sensitivity analyses where we're leaving out certain samples to make sure that they aren't the things that are driving that positive result. So, going back to the question, repeating the results a few times is really just doing the same exact analysis and then picking a representative run. But what we're trying to get at is this broader idea of whether or not that result is stable given the stochasticity of the algorithm. And then what you can do by repeating it more is to dig into that result a little bit more and understand what's driving that result and make sure that you really believe it.
Ah, cool. So next one here, Peter asks, you mentioned Cytobank has normalization as part of Citrus. Does that help with batch effects?
That is a great question. So the normalization that is built into Citrus is intended to help with differences in the expression of markers in different channels. So like Geoff was mentioning earlier, if one channel is super dim and one's really strong, it will help kind of equalize and handle those differences, but it is not designed to handle batch effects or help with that. So in that situation, you would want different normalization workflows, which we're happy to talk to you more about if you're interested in that.
All right. Let's see. James asks, do you have practical advice on translating marker profiles for clusters from the PDFs?
Geoff, help me brainstorm about that a little bit.
It may just be, and it depends. We can certainly follow up with this, but translating marker profiles for clusters from the PDFs, it may just be kind of what their phenotypes are, and if you say, all right, so I have one cluster, and it has these this level of expression on these different surface markers. You can obviously there's that series of histograms that tells you what the histogram or what the levels of marker expression are for all the different markers you used to generate these clusters. However, you can also, if you're curious, like I mentioned before several times, you can export that cluster and then kind of look at it on a series of two-dimensional dot plots to see how it compares to kind of like your normal gating hierarchy that you might have generated manually. But again, we can follow up with that on a separate email.
The other thing I think that I would add to that is, once you get to that experiment where you've exported your significant clusters from Citrus and you're looking at those, you can do any number of different things in that experiment that you would normally do. So you can export statistics from those files on the different marker profiles, and that would be like within the significant clusters now. And then you can also make things like heat maps to look at marker profiles there within that exported Citrus cluster experiment.
Yeah. Victor asks, if you run Citrus three times, the results differ. How do you report that? Would you say in 2 runs, cluster X was significantly different between the groups but failed to be in 1 run? Or would you only include data that returned significance in all of the Citrus runs?
So typically we interpret that to mean that if something is significant, in only 2 of your 3 cluster or Citrus runs, we interpret that to mean that it's not actually significant, that it's a false positive result of the stochasticity of the algorithm. However, it's always a good idea to state what you did and kind of summarize those non-significant results. But when you're summarizing the significant results, we wouldn't include that typically.
So Alexandra asks, can you explain in the simplest possible terms exactly what the values in the CSV files represent for both the median and the abundance analysis?
Yeah, so for the medians, that would be the median expression of whatever marker you're looking at in that particular cluster. So the cluster is a cell population that's defined off of the surface markers that you chose for clustering. And then the median is the median marker expression of that marker that you're looking at in that population. And then for abundances, it's the abundance or percentage of cells in that significant cluster that we're looking at out of the entire cell population that you started with. So out of whatever population you pre-gated to that you put into your ViSNE and then into your Citrus.
We have one here from Sam. He asks, what should I do if I have Citrus groups that have different numbers of samples in them?
That is a good question. So unbalanced groups is something that you need to worry about or think about, I think, in a couple of different contexts. I would say it matters a little bit more generally for predictive modeling. And so what you need to understand about that is that when you're building these predictive models where you're answering the question of what's the fewest number of markers that I need to best predict the differences between my groups, what Citrus does is chooses those models based on looking at the overall accuracy. So how well can it predict each of those groups that you input? And what that means is that if you have unbalanced groups, the algorithm might not necessarily be incentivized to choose a model that's actually discriminating between your 2 groups. So for example, if I have 100 samples in total and I have 99 samples in group A and 1 sample in group B, the model could actually achieve just a 1% error rate by calling everything group A. And that wouldn't be very useful scientifically because it's not going to be using any markers that actually discriminate those groups to make that prediction. So that's kind of a thing that you have to keep in mind when you're looking at unbalanced groups with predictive models. With correlative models, you have to worry about it a little bit less. I think the thing to keep in mind there with having unbalanced groups is that if you have a small number of samples in your smallest group, the differences that you find might not be consistent if you were looking at a larger sample size. So that's just like with any statistical test, where when you have a small group, you need to worry about how consistent your results are.
All right, great. Well, I think we're just a little bit over time, so we're gonna cut it off there. But there are more questions that you all have asked, so we're gonna follow up with you individually offline. I'd like to thank all of you for attending. I'd like to thank Kat for taking the time to come in and answer some of these questions. Again, thanks so much for time and attention, and we'll catch up with you next time.